Hello, everyone, and welcome to today's broadcast, Experimental Design Best Practice for Multicolor Flow Cytometry, presented by Carol Oxford, Field Application Scientist, Thermo Fisher Scientific. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit www.thermofisher.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, click on the arrows at the top right-hand corner of the slide window. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Ms. Carol Oxford. Carol Oxford is currently a field application scientist with Thermo Fisher Scientific. She has worked with FlowJo LLC and was one of the founding partners of Xcyte, a flow cytometry education company. Ms. Oxford, you may begin your presentation.
Thank you so much, Xavier. I really appreciate the opportunity to come talk to you about flow cytometry. I've done this for quite some time. I was also the UC Davis flow cytometry core director there for about 25 years. So I think flow cytometry is one of the most elegant technologies in science. When I did flow cytometry, multi-parameter meant two fluorescent parameters. And now the people at the cutting edge of flow cytometry are doing 32 fluorescent parameters. So it's changed quite a bit during that time. But I'm really thankful to all of the mentors that I had. And so now I have a lot of experience and hopefully you'll find some information here that's helpful to you as you design your multi-parameter panels. I put my email address here on the slide so that you can contact me after the seminar if you want any information that I give here. You can also email me about other topics in flow cytometry, and I can give you some resources as well in the seminar. So let's just talk about flow cytometry in general. There are advantages and disadvantages, of course, to every technology that we use in science, and there are a lot of advantages to flow cytometry. One of the things that I like the most about this technology is that we get a lot of information on a per cell basis. We use the populations of cells that have a lot of heterogeneity, but we're doing this on a per cell basis. It's extremely quantitative. We look for statistics like population frequency and expression level on a large number of cells. We can look at tens of thousands or even millions of cells in an extremely short period of time. And we can generate this data very quickly. Of course, there are some limitations of flow cytometry. Despite our best efforts, the cells must be in suspension. And those of us that have certainly run a core facility or run our own data, we realize that putting clumps of cells or putting pieces of tissue in a flow cytometer quickly ends that experiment. And we spend a lot of time fighting with the flow cytometer. So we have to get the cells in suspension. We have to make sure they're filtered, or we use enzymes to make sure that the cells pass through the flow cytometer. The other limitations are we get no spatial information unless we use a complementary technology like microscopy or an image stream, which gives us spatial information about where the cells are. To flow cytometrists, cells are an electronic pulse. We don't get any information about where the fluorescence is inside the cell. And one thing that's interesting about flow cytometry data is it's very easy to misunderstand the data. And I get this all the time from clients and students who tell me that, you know, I've had 30 years of experience, so they want me to look at their data or they want me to analyze their data. And what I want to present today is that you don't have to have 30 years of experience to get good flow cytometry data. This data is not subjective. It's very important to have the adequate controls to understand a little about physics and a little about antibodies, and to do a very careful analysis. People that are just starting out in flow can do the analysis the same way somebody that has 30 years of experience. It's just really important, and sometimes, in fact, most of the time, you'll have more controls than you will have samples. So it's just really important to follow some important steps to get really good flow data, and we'll talk about most of those here. So before you panic, this slide is extremely busy, and it has a tremendous amount of applications in flow. I know there were a lot of times in my career that I thought, well, we're pretty well done. We have a number of applications, but we're kind of stuck. And then fluorescent proteins came to the table, certain dyes came to the table, and probes came to the table, and all of a sudden we were off and running again. So I'm certainly not going to go over any of these applications in particular, but I wanted to say that if we can develop a fluorescent probe or a fluorescent marker, the list is almost endless in the applications of flow cytometry. So let's talk about flow cytometry best practice. These are the things we're going to talk about today. They don't encompass all of flow cytometry best practice, but they're certainly important to panel design. The first thing we're going to talk about is how do we know the machine is performing optimally? Certainly the manufacturer of flow cytometry instrumentation is going to develop a protocol to give you an idea of whether the machine is performing optimally. And we're also going to talk about what you might want to do above and beyond this level of testing to make sure you're going to get good data. We're also going to talk about how to choose fluorophores. It's very important to understand some of the characteristics of fluorophores that might make you choose one over the other and give you an advantage in panel design. We're certainly going to talk about titration. This is one of the most important and most simple concepts that will give you good data and make panel design much easier. And then, of course, getting along is important in the workplace, it's important in relationships, and it's incredibly important in panel design. Not all fluorochromes get along, and we'll talk about which ones do, which ones don't, and how you can get them in a panel together. And then, of course, control issues. Me? Well, I didn't think I had control issues, but when you become an expert in multicolor panel design, you're going to have control issues. You're going to run a lot of controls, and you're going to want to know what each one is for, and you're going to have a lot of them. So we talked about that already, and you're probably going to be sick of how much I talk about them in this talk. So bear with me. You'll still love flow cytometry by the end of the talk. I do, but we do have a lot of controls. So one of the most important things to understand, of course, right, and when you're using any machine, is what does it do? Now, I'm not going to get into the heart of physics because I shouldn't say I don't like physics, but I'm not a physicist. So I understand the least I need to know about physics to understand how the machine works. So in the top upper left corner of the slide there, you'll see what happens when you open the machine, the Attune NXT flow cytometer, which I represent as the field application scientist. So here you can see the filters of the machine, and you can see the lasers and some of the optics. So it looks pretty complicated, but it all boils down to the detector. What is the cytometer doing? It's counting photons. So we measure the photons that are excited when the laser excites the fluorochrome, and the fluorochrome emits photons. The photons hit the photocathode at the front of the PMT. Those photons, the electrons that are now knocked off that cathode go through a dynode cascade, and they're amplified. They then are emitted off the other side of the PMT, and we can measure those with an electronic pulse. Very, very straightforward. So all you need to know is that we quantify the photons, we measure the electronic pulse, and there you go. So if you know a little bit about physics and a little bit about photons, you're now a flow cytometry expert. So when we started in flow cytometry, we used analog instruments. We didn't know a lot about PMTs, or at least I didn't, and we had a 4-decade scale to graph our data. So we could walk up to an instrument, we could take an unstained sample, put it in that magic place that we know is no longer magic, that first log decade on our scale. We knew that most of our fluorochromes, most of our samples would fit in that space, and maybe sometimes we had to turn the voltage down, but otherwise we got pretty good data. Then we started using analog to digital hybrid machines and then fully digital instruments. And we saw all sorts of things happening. We saw that if we crank the voltage up, there were populations appearing that we didn't see before. And we saw our background increase sometimes. We saw all sorts of things. So we realized that we needed to understand a lot more about PMTs. We needed to set voltages that were appropriate for our experiments. So we needed to do some optimization that's now been called either a voltage walk or a voltration. Voltration sounds really cool. If you print it on a t-shirt, it makes you very popular at parties. I'm just kidding. I also find my jokes really humorous. So what does a voltration do? It assures the best signal-to-noise detection, which is what we want to do. Our data is important to know the difference between background and negative and positive. We also want to assure all the measurements are being made in the linear range of the detector. So before, when we only had 4 decades, and now we have instruments with 5 decades, 6 decades, and 7 decades of range. So we need to know how many decades of real data we get from the electronics. So decreasing the voltage below the minimum voltage that we find in a voltration, the resolution of dim populations may be lost. Increasing the voltage for a given PMT above this voltage gives no advantage in population resolution. So I get a question all the time also from customers. Why not just put a sample on and increase the voltage until the positive is almost off scale, which is really what we first did. If a low voltage is a problem because we're detecting the noise of the instrument, why not just crank the voltage up? Well, then we realized that that instrument noise can also become a problem, and we get spillover spreading from the machine. So I'll explain this as well. So this is one way to do a voltage optimization. So we used here our ultracomp beads, and I'll talk about those in a minute, but basically they're antibody capture beads, and they contain a binding bead and a non-binding bead. These beads bind to the variable region of your antibody, so you can put your antibody in the tube, put the beads in the tube, and they have 2 populations. They bind to the variable region. So here you can see at the PMT voltage of 1 volt, which is basically off, we haven't applied enough voltage to the PMT to get a signal. And here you can look that there's basically only 1 population. Both of those beads are not being excited enough to see. And we start walking the voltage up, very straightforward. And as we get 2 populations to appear, and then the beads start to go off scale, we lower that voltage difference to about 25 volts. And this is the detector here. If you can't see the slides very well, the detector is the BL1 detector on the Attune, and that is excited by the blue laser, and it emits in the emission filter there is 530/30. So we're collecting green excitation off a blue laser. So if we do what's called a concatenation in FlowJo, you can look here and see some interesting things about the data. You can see that down at the low end at 250 and 300 volts, the data is not linear. You can see the data starts to become linear when we hit 350 volts. So we know we want to be above that range. You can also see that maybe somewhere around 375 volts, that background starts to spread. That's going to become a huge issue when we start using multicolor panels. So here, how would we decide what voltage to use? Well, I can pick a magic red arrow there and I can tell you that's what voltage I've picked, and I'll tell you why in a minute. So here's a voltage optimization that's been done on an Attune NXT, and I've walked 5 PMTs off our blue laser, 3 off the red, and 3 off the violet. Now you can do this on your machine as well. You walk each PMT individually, and this is what you get when you visualize the concatenation in FlowJo. So what in the world do you do with all that data? Well, there's a number of things you can do. So what we did, and we published, or we submitted an abstract at Cyto, and I can certainly send that to you if you want. It will also be published in our bioprobes, and I can send you the link to that as well on our website. So here is the data looking at, again, that BL1 PMT and also the YL4 PMT. The YL4 PMT is yellow excitation, and the channel that you would, sorry, I don't know the filter there. I can, that's the excitation of PE size 7. So that's long red emission off the yellow laser. So we looked at several metrics here. The stain index, which I'll explain in a minute, give you the formula for that. You can Google Marty Bigos and stain index for the publication on that. We also looked at separation index. We looked at robust CV of the positive signal. We also looked at 2 times the standard deviation of the negative signal. There are a lot of metrics you can look at here. Basically, what you want to see is you can look at where the robust CV starts to decline and flatten out, and then you can also look at where the peak of the stain index is and where that starts to flatten out. So here for the YL4 PMT, for example, that starts to happen at about 400. So these are some various methods and metrics you can look at for the voltage optimization. So if you look at these graphs, you could certainly argue that there are several voltages that might work, and that's a valid argument. The next step in this process is to take your data, run them at voltages here, a minimum voltage, say 400, 375, 425, and see how this affects your data. Look at some data with some low expressing markers and see if it makes a difference. The real point of this optimization is to find a minimum baseline voltage. You may adjust those voltages up or down, but probably not. This is a good place to start every experiment. Here's another option for your voltage optimization. Here you have a hard dye bead. The bead that I used for this optimization is the Peak 2 of the Spherotech 8-peak bead set. This is a hard dyed bead. It is a single peak, so I don't have 2 peaks here that I have to set a gate on, and I did some very simple metrics. I used the robust CV and the robust standard deviation, and I looked at those graphed simply in Excel, and I looked at the range. I didn't look at the inflection point. That's also an interesting point, but I looked basically at the robust CV, where that leveled off, and I looked at where the robust standard deviation started to rise. So for example, for this PMT, I would probably pick a voltage of about 350 for my minimum baseline voltage. And the reason I would pick that voltage is I get a good flat CV, so I know increasing the voltage there would not give me any advantage, and I have a very low contribution of the electronic noise measured by the standard deviation. I've done this on several occasions, on multiple occasions with multiple machines. I've also run data here, and that suggests that I get a very good signal, and I also get good data here. So the next step that I do in this optimization is I take another hard dyed bead, for example, the Peak 5 bead, and I walk my instrument for all PMTs doing this optimization. When I find those voltages, I make a histogram with all of my PMTs. Then I run the voltages from the optimization, and I run my slightly brighter bead, my Peak 5 bead, at all of these voltages, and I set a gate. And I run this every day, and I use the voltages every day for my experiments. That way, if my instrument changes, if anything changes in my experiment, I can say to the FDA or to the reviewer of a paper that I have measured the same fluorescence every day. So this is a great way to not only make sure that your instrument is characterized, but make sure that you are characterizing your experiment as well. So let's review the voltage optimization. We want to optimize our PMT voltages so we have a minimum voltage where we are always detecting a voltage or detecting our cells and our signal above background. If we do need to go below this minimum, and why would that ever happen? If I put on my cells and I realize that now my positive signal is off scale, if I haven't titrated, or maybe I'm using GFP and I just can't get that signal on scale, it's the most important that you have to get it on scale. So you're going to lower your voltage. If I have to do that, I know that I may be sacrificing signal to noise, but at least I have an understanding of that. And with GFP, if I'm only looking at something bright, signal to noise may not be my most important consideration. If I raise the voltage, there's no advantage to resolution. So I don't want to change these just for resolution, and optimization is very important, so just get it done. If I do this on an Attune NXT with the hard dyed beads, it takes me about an hour. I throw the numbers into Excel, and it's very easy. Okay, so now we have our flow cytometer ready. What's next? So there are many ways to do panel design. There's an excellent paper by Yolanda Mankey and Mario Roederer. I believe it's from 2010, but if you search panel design and those two names, M-A-N-K-E and R-O-E-D-E-R-E-R, you can find the paper, and it talks about ranking your markers. You want to choose the markers which best answer your research question. They rank them primary, secondary, and tertiary. I find that very helpful. It's not engraved in stone, but it's very helpful because, again, I use this little rhyme. The more colors you use, the more sensitivity you lose. It's counterintuitive, but the more fluorochromes you add to a panel, you encounter a concept called background expansion because of spillover spreading. And this is a problem of physics. It's not something that compensation corrects for, not something we can avoid. So if you can answer the question with less markers, it's always an advantage to do so. But of course, nobody wants to do that, right? We want to use more markers. So we'll talk about how to do that. You're going to want to titrate all your reagents. You're going to want to screen for the best ones. What are the best ones? Well, of course, they're the ones that separate the population best, but also sometimes the same reagent conjugated to a different fluorochrome is going to give you a different staining pattern. So you want to test all the combinations that you can. You want to troubleshoot for issues with sensitivity. I'll give you some tips there. And then you generate your data. So of course, this goes back to high school science, right? Or maybe even elementary school science. We determine a biological hypothesis. Which antibodies do we need for markers of interest? You're going to do a literature search, and there's a really helpful source that you can do this. They're called OMIPs. I'll talk about that in a minute and show you where you can find them. But the most important thing is that panel design is iterative. Predicted panels are exactly that. They're predicted panels. And no matter how much expertise you have, I can give you a predicted panel, but I can't tell you if it's going to work or not with any confidence. I used to teach week-long courses in flow cytometry, and every single class, there'd be people disappointed at the end that I couldn't hand them a panel that worked. I certainly can hand you panels that I've done. I can give you panels in the literature that other people have done, but they have a successful chance of working, but you are not going to do, well, unless you want to repeat their work, you're not going to do the same thing on the same machine with the same cells. So you're certainly going to have to do this in an iterative manner. The idea is that you're going to do a backbone panel that always works, and then you're going to add antibodies and add markers and see if that backbone works until you can add as many markers as you need. But there is a lot of help out there. The flow cytometry community is really, really collaborative, and OMIPs were designed, or actually introduced by the reviewers of the journal Cytometry. So I think there are more than 50 now. These are open source, and they were designed because no matter where you publish a paper, you can never get all the information that you need from the journal's methods section. So let's say I want to publish a paper in Nature, Carol's Cool T Cells Kill Cancer. So unfortunately, you can never get all of the important details out of the materials and methods section. So I can get another paper, a 2-page paper called an OMIP, Optimized Multicolor Immunophenotyping Panel, and I can get that from, or I can publish that in Cytometry. So this gives all of the detail that flow cytometry nerds want to see, right? You can see important things like the gating strategy, the clones, the panel design. Did I have any difficulty with certain antibody combinations? Here you can see in the gating strategy, I don't use a forward and side scatter gate to determine any of my cells of interest. I just use it as a debris elimination gate. I gate first on my doublets. I gate second on my dump channel and viability dye. I use different types of plots. I use maybe a plot that shows me some backgating that I did. So it's really, really critical that you can use these as a reference. We also keep track of these on our website. You can look at our flow cytometry resource library, which has a tremendous number of resources for both people that are new to flow cytometry and people that are very experienced. We have flow cytometry application notes, techniques. We have videos so that if you are new to flow cytometry, you can figure out how a flow cytometer works. But we also keep track of all the OMIPs here, and so that, because if you Google them, sometimes only a few of them come up. I really appreciate them because when I studied T cells, they were just activated or producing cytokines. Now, people ask me about panels for T cell exhaustion. Well, they weren't exhausted when I was studying them. You guys have the hard work to do, right? The old people in the field, we did the easy work. So these will really help you with your research. So I talked about the stain index. This is the formula for stain index, basically the median of the positive minus the median of the negative over 2 times the standard deviation. It's a metric that allows us to look at the separation of our populations. There are many of them out there, but this is one that we would commonly use. So antibody titration. People always ask me what's the best way to titrate an antibody. Any way you want to, right? There are a tremendous amount of protocols out there for this. One of them that I like is by Ryan Duggan. If you Google Ryan Duggan and titration and Alexa 488, he has a great protocol that actually tells you the amount of PBS to put in a well when you're titrating your antibodies. This is a titration that was done by one of our scientists. It looks at serial dilutions of a 1 to 2 concentration of antibody, and then the graphing of the stain index. So this is where there's a lot of mythology about antibody concentration. So here's the graph of the stain index that you can see. And when I was new to flow, I would have said, well, I would look at this and pick one of the higher concentrations of antibody where that stain index flattens out, because I want to be at saturation, which might be the concentration here. But then now I've learned that you can use a concentration here. There's a difference between a saturating titer and a staining titer. The one thing you're trying to do here, or maybe you're trying to do, is to make sure that in the population that you're trying to detect, you're detecting the percent positive. Now, if you're trying to see the population shift or the MFI shift, you're going to want to use the saturation titer. That's a different case. But most of us are just trying to say, I have 20% of these or 50% of those. So in that case, you may want to use a saturating or a separating concentration. The saturating concentration, if you increase the antibody, you only increase the nonspecific background. People always ask me, well, why can't you just wash more? Washing does not eliminate nonspecific background. At least it doesn't very well. Washing is a great way to lose cells. You usually lose about half of them in every wash. It does eliminate excess fluorochrome, but that's really not the important thing that you want to do when you're titrating and staining. A separating concentration gives you good separation of your cells, it reduces your spreading error, and it saves you antibody. So you can often use the separating concentration when you're only looking at the percent positives, which is really what the vast majority of us do when we're doing immunophenotyping. So let's talk a bit about spillover, compensation, and spread. These terms are thrown around a lot in flow cytometry. They're sort of the language that flowers speak. You'll also hear the term spillover spreading. So bear with me a minute as I talk about something that's not on the slide here. When I talk about compensation, I often use FITSI because it's a fluorochrome that we're all really familiar with. And when I ask what color is FITSI, you say green. Well, I just set you up for failure, right? Because it's a trick question. FITSI is not green. We think FITSI is green because we've been scammed. FITSI is green, or we think it's green, because we look at it under a microscope. And we have a filter on that microscope that only allows those really energetic green photons back to our eye. So the truth about FITSI, I feel like making a Netflix documentary. The truth about FITSI is that if we didn't have that filter, the emission spectra, and here you'll see the emission spectra of PE Sci 5.5 on your screen. I'm sorry, maybe it's Per-CP Sci 5.5. Yes, it is. So an emission spectra is a probability curve. So going back to FITSI, if I hit FITSI with a blue laser, the chances of FITSI, or the probability of FITSI emitting green photons is very, very high. But FITSI also has a very good probability of emitting yellow photons, or orange photons, or even red photons. And I have a flow cytometer that very well detects yellow, orange, and red photons. So, but it's very easy for me to determine where those photons are coming from if I have a single stain for every color that I use in my experiment. So we now use compensation, which is an algorithm, to separate the emission of these fluorochromes and visualize them in multidimensional space. And that sounds really tricky, and the math might be too complicated for me to do in my head, but it's very, very easy in practicality. So here is two fluorochromes that we use, Per-CP Sci 5.5 and APC Alexa 700. And here's the example of where the photons are that are emitted by one and detected in a neighboring PMT. So when I use a single stained per CP Cy 5.5 sample, all of the cells or all of the beads, whichever this is, are detected in the detector, the primary detector where I want to make a measurement. They're also detected in this secondary detector where I'm going to eventually make another measurement. And this cell gets a number here, and it also gets a value in this detector. So compensation just uses an algorithm to force the median of this value on the y-axis to be the median, to be equivalent to the median of this on this axis as well. We could have chosen another algorithm to do this, but we chose compensation. So compensation allows us to visualize the effects of spillover in multidimensional space. Sounds complicated. It's not. It just uses a vector and the slope of this line to do the calculation. So here we have a median of 1,277 and 60.2. I'm sorry, we have an MFI here in this, on this axis. We use compensation in our single stain sample to adjust these medians to be more equivalent, and we have a fully compensated sample. Now, when this cell gets a value here and this cell gets a value here, I can't change that value. I can only bring it down to the lower end of the log scale, and now I visualize this spread. It's not visible on this because, it's not visible here, sorry, because it's on the high end of log space. It's much more visible here. This is what is called spread or spillover spread. I don't really like those terms, but we're kind of stuck with them. So that's what you see here. So this is an important example looking at uncompensated and compensated data. Usually, we can recognize that the plot on the left is uncompensated by the strange diagonal. And on the right, we only know the data is compensated because it's marked uncompensated. We really don't have any way to statistically determine that those medians are equivalent in the compensation controls. But it's important to know that we've used the right compensation controls, and we don't know on the left that the population that is dimly stained for C11b and brightly stained for C11c is indeed double positive. So we want to make sure and use the right compensation controls to make sure we have good data. So the process of compensation is very easy, even though the math is kind of complex. So compensation beads are really the easiest way to do compensation. They work for all lasers, and depending on the manufacturer, they work for every antibody. The beads that we carry, the eBio ultracomp beads, work for mouse, rat, or hamster. The ABC beads also work for rabbit antibodies. The features that are nice about the beads, the unstained bead fluorescence is very similar to most cells, and they give you a bright stain and a clear separation. So the rules of compensation, of course, are that you have to have a single stain for each fluor in your sample or in your panel. You need to have a negative and a positive either in the tube or in the algorithm, so you can use an unstained. It used to be important that you had the sample as bright or brighter, but when I've talked to experts in the field like Dave Parks, who writes the compensation algorithms in the software, he said the algorithm should calculate that math for you. I think it's still probably important to have a very bright and well-separated population, and the beads do this for you. It's helpful certainly if the target is not highly expressed on your cells or expressed by only a few cells. So for example, if you're using a cytokine or FOXP3 or something that's not well expressed, these compensation beads work really, really well. So now that we've got our compensation controls done, our instrument voltages are set, we want to talk about some basic strategy for fluorochrome selection. So we wanted to save the brightest fluorochromes for our dimmest markers. So how do we know that our markers are dim or bright, or our fluorochromes are dim or bright? Well, I'll talk about that on the next slide. We want to save our important targets, which we ranked with the paper that I discussed. So we want to save these for the brightest fluorochromes: APC, PE, Alexidyes, super brights. These are the worst resolved. They have low expression or poor antigen access, or they have an unknown expression level. If I want to look at antigen X, I may not know what the expression level is. Then I want to minimize the spillover. So I think about this as part A and part B of panel design. I like to have a spillover spreading chart, that's part B, and then I like to have a stain index chart, that's part A. So I want to look at stain index and spillover spreading. So let's go to the stain index slide. Here's an example of the ranking of fluorochromes by stain index. So this is one that we did at Thermo Fisher. We took freshly isolated PBMCs and stained them with anti-human CD4 conjugated to various fluorochromes. Now this, of course, will vary manufacturer to manufacturer. It will vary instrument to instrument. So what we've listed here is the fluorochrome, the laser, the filter, and the stain index that we got with the Attune NXT flow cytometer. You can certainly use this on your instrument. These things will be relative instrument to instrument. It is best to use it on your instrument, and certainly different manufacturers will have this for their instruments and their fluorochromes. I like to have this printout in front of me so I can see the brightness of fluorophores. This also depends on several things. Fluorophores are bright because they're quantum efficient. For example, FITC is very quantum efficient. When you hit FITC with a lot of light, you get a lot of photons. But FITC, unfortunately, can be a problem if you're looking at an autofluorescent cell line, because you also have, of course, autofluorescent and autofluorescent proteins emit in the green. So the brightness of this only accounts for the stain index and not necessarily for the autofluorescence. So that's another thing you'll want to consider. But this is a great chart to have as you're doing panel design. So here we'll start out talking about spillover spreading. So we talked a little bit about looking at fluorochromes that have emission spectra that overlap. So on the left, you'll see an example of CD3 APC Electrofluor 780 and CD56 APC. You can see that those emission spectra overlap a bit. And if you use these on cells that are co-expressing the same markers, you can certainly see that the pattern that you get, even if you used an FMO, it'd be very difficult to see how to quantitate those populations. If we separate those co-expressed markers onto fluors like eFluor 450 and APC, we can see a much better staining pattern that gives us a much better idea of how those cells are co-expressed. And this is a classic example of two fluorochromes that are used quite often in panel design. So here's an example of PE into Per-CP Sci 5.5. PE is a very, very bright dye. And it's often that manufacturers, when they come out with a new antibody, the first thing they do is conjugate it to PE. And I'll tell you that PE is possibly the worst place to make a sensitive measurement in your cytometer. Now, PE is very bright. We use it all the time. But PE is also the place that accepts a lot of error from the other fluorochromes that you use on your cytometer. And I'll show you why. So here we have a PE single stain, and we're measuring that in PE and in Per-CP Sci 5.5. So again, as each cell goes through the cytometer, it gets a value in every detector. When we compensate that data, you can see the spread of this data. So compensation lets us visualize the spillover and the spread. So if we used a gate on the unstained population, obviously it looks like there's a population of double stained cells on the right. Those aren't double stained. We can certainly see that. We would have to use an FMO. But these cells are definitely there. So there's an easy way to do this in multicolor space. This is a neat trick. So here, instead of thinking about how bright our stain is, right, which is part one of panel design, the stain index, now we're going to think about if we have to have, for that Nobel Prize or that paper in Nature, we have to have a Percy PSI 5.5 or a FITC antibody. And here's the FITC antibody I absolutely have to have. And here I'm looking at that antibody in FITC and in Percy PSI 5.5. Now, I add more antibodies to this panel. I've added the two antibodies that I cannot live without, and I've added APC size 7, BV421, PE size 7, and the other antibody that I have to have in my panel. Now, with all of these dyes in the panel, I can still use the two antibodies I need without any problem. You can see the background expands a bit from spreading of the other fluors, but no matter where I set a gate, I can easily determine the population percentage of those two dyes. Now, I add PE, BB605, and BB711, and now I have a problem. With the two antibodies I'm using, the FITC and the Percy PSI 5.5, if the staining is as bright as it looks here, they're not a problem. But if the staining is dim, now I have fluorescence all the way out to the third log in the Percy PSI 5.5 channel. The FITC channel is virtually ineffective. FITC is one of those dyes that plays nicely with others. It does not accept error from any of these dyes, certainly. But the Percy PSI 5.5 channel, there's background expansion. And right now, all I know is that it's from PE or BB605 or BB711. Now, certainly, the more experience you get with panel design, you'll know where it's coming from. But this little trick can tell you exactly where it's coming from. So here, I can look at every single stain sample, and I can look at it in my FITC channel. And like we saw before, all of these single stain samples look fine when we detect them in the FITC detector. Now, when we look at all of the dyes, single stained and concatenated in FlowJo, we can see exactly where the problem is, PE and BB711. When I add these two dyes, they contribute quite a bit of background expansion to the Percy PSI 5.5 channel. So I know that if I'm going to use an antibody in Percy PSI 5.5, it has to be brighter than that 10 to the 3 point, because if it's dimmer than that, I just won't see it in multicolor space unless I use it on different cells than I'm staining with the PE and the BB711. And our new super bright dyes that also emit off the violet channel would have the same characteristics because they have the same emission or very similar emission spectra. So here, for example, are all the same dyes in the BB711 detector. So you can see Percy PSI 5.5 is going to be causing some background issues for BB711. And here you can see all the dyes that cause background issues in PE size 7. Again, PE size 7 is very bright and often used in multicolor panels, but you can see it's not a place that you would necessarily want to make a dim measurement. So these are very important measurements to make and understand which dyes cause contributions and which dyes you might want to either keep separate or save for your brightest markers. Let's just do a quick spreading error review. These are basically caused by photon counting statistics. I like to think of photons like kids. Basically, we really like energetic kids. We like blue, green, violet photons. They have tremendous amount of energy. when they hit that cathode, they knock off lots of electrons. And as scientists, we like ends of lot of numbers, right? We can count those. We get great statistics. We get very low variation. And unfortunately, the red photons are like teenagers. I love my children. I raise them through their teenage years, but they're a very bad investment. You put lots of energy in, you never get that energy back, right? There's loss through rotation, translation, heat energy. And when they knock off electrons, sometimes they knock off a little, sometimes they knock off a lot. And when we count them, there's just this spread, right? We can still use them. There are advantages to using them. But we need to understand that we get the spreading error, and there is a way to predict it, and there is a way to use them wisely. So here's an example of a great paper in Mario Roederer's group, and this is a calculation of some spillover spreading matrices. Now, this is another paper that is very complicated, and if you have to do the math by hand, I don't envy you. We used to do this on all of our machines in the lab, and it took a lot of time, but it's very valuable data. Here you can see where the fluorochromes don't get along and where you're going to have issues. These are on LSR2 instruments and looking at Q dots and some of the other fluorochromes. So this is an example of a spillover spreading matrix that I generated in FlowJo with some of the scientists at Thermo Fisher. So this is a very easy thing to do in FlowJo. So you take the data of single stained, these are CD4 on an Attune NXT V6 configuration, and these numbers give you some warning signs based on the red color. So for example, this is VL5 and RL2 on the Attune NXT. This is super bright 702 and Alexa 700. So these emit in the same channel off of different lasers. I'm sorry, they have a similar excitation spectra off two different lasers. So you can see where they might have an issue. And so this is just a little bit of a warning to say that you're going to have to be careful if you use these two together on the same cells. So this is really helpful. You can do it easily with single stain cells. This is with CD4 and super bright dyes and several different fluorophores on a Attune NXT. So this is really helpful and very easy to do. You can do this for your machine. It doesn't take long, just run the single stains, throw it in FlowJo, and here's a button you click on for the spillover spreading matrix. So we'll talk a little bit very quickly about staining controls. This is in the literature, and there's a lot of great articles here, so I won't go too heavily on them, but there's some very important controls that you need to do when you're doing multicolor flow cytometry. And the number one control that I find people are not using as much as they probably should are fluorescence -1 controls. These controls are not compensation controls, they're gating controls. And then we'll talk a little bit about the rest of the ones on the list here. So fluorescence -1 controls contain all of the markers except the one of interest. So if I want to do a FITC FMO control to determine what's positive and negative for FITC, I want to use all the other fluorochromes in my panel except for FITC. They help us control for the effective spread. They allow delineation of positive from negative, and they're important for low density or smeared populations. So here's a couple of examples of how a fluorescence -1 control is used in gating for flow cytometry. You can see on the left, in both cases, we've set a gate on the unstained. And on the right, you can certainly see that that gate is not appropriate for the full stain. And you want to move the gate up, but you certainly can't convince a reviewer that Carol said I could move the gate up. Unfortunately, that doesn't get a paper into a journal. I wish it did. But if you run the FMO, you can see that that gate definitely defines the population and describes that spillover spread that we see there. Setting gates is not arbitrary, and I can certainly not set gates any better with 30 years of experience than you can. I use the proper controls as well. Don't want to rely on the unstained. And certainly people ask, how many FMOs do I need to run for every experiment? So what I do is, if I'm doing a 14-color experiment, I'm going to do 14 FMOs with that first experiment. And here, for example, in the first panel of plots that you see, the PE-Cy7 and the CD45 PE-Cy5.5, I would not need to use those two FMOs after the first time I run them, because no matter where I set those gates, I can get a good read on the separation between those two populations. Once I've set the FMO and I realize where I need to set it, I can tell a reviewer, I ran these FMOs the first time, I got good separation, and I didn't have to run them the next time. In the bottom two panels, I probably would not need to use the CD8 FMO, but I would certainly need to use the FMO for PE, because there the antigen is expressed on a continuum, and I would really need to set that FMO every time, because moving that gate very slightly in either direction is going to change the value significantly. And here again, also, I probably wouldn't even use quadrants for this gate. I would probably set them as squares, because quadrants really don't describe flow data very well. So here again, you have controls based on what you're going to do to your cells. If you make changes, or if you treat cells with radiation, or you stimulate them, or you kill them, or there's something that you do to your cells that create a change in your cells, there may be changes that affect your fluorescence, that affect your scatter, that affect the stickiness. So you want to do as many controls as you can, especially in the beginning of an experiment, so that you can see any of these changes that might occur. Isotype controls are very controversial in flow. They're not often used anymore. There's a tremendous amount of literature out there on them. If you have to use an isotype control, you want to throw it into your FMO at the titration that you use your antibody. And really the only thing that an isotype shows you is that you have a titration issue. It shows you that you have a nonspecific binding problem. So it's really something that you might want to consider only using in your FMO control. Viability, it's very important to eliminate unwanted cells. Dead cells are sticky. It's very important to use a viability control in every sample. Dump channel, not necessary, but often very helpful. You can eliminate unwanted cells from analysis. This is being very helpful in things like stem cell analysis. Stem cells are very rare. We want to make sure we look at undifferentiated cells. So you can add differentiation markers all in the same channel. You can use them in the same channel as your viability dye and gate them out. So here's a slide that shows the Amine reactive dyes for dead cell identification. We have 8 different options at Thermo Fisher. These are great. We used to use EMA, which required ultraviolet light fixation. And when these came out, these were fantastic. Live cells have some amines, dead cells have many more. So you can take these dyes, slot them into a place in your panel where you don't have another reagent, gate them out. We have ABC amine reactive compensation beads, so they're really fantastic for use in your panel. We also have a number of impermeant nucleic acid dyes for dead cell identification. These are very bright, come in a number of colors that you can also slot into your panel. These are for use for live cells. The others can be also, the amine reactive dyes can also be used in live cells or in fixed cells. So here's an example of the importance of using a viability indicator. So forward and side scatter, we know are not to be used for dead cell indication because they really are not a good indicator of viability. If you gate here on the upper left panel and you see a forward and side scatter gate, this is using log side scatter as the Perfetto and Herzenberg and Roederer lab often do. You can see here that the live cells and dead cells that are determined by scatter, if you gate them separately, you'll get a very, very different phenotype. And you'll still have dead cells in the final analysis that look like CD107 positive cells. And so it's very important that you gate them out in the beginning using a dead cell indicator. So here's a slide just looking at the differences that you'll get when you exclude dead cells, when you use a dump channel, and when you exclude doublets. So if you're looking at rare cells, or if you're looking at any cells, it's really important to exclude the cells that you need to exclude in flow cytometry. So in summary, know your instrument, know the configuration, know what your performance test that's determined by your manufacturer, know what the results give you and how your instrument's performing, do a voltage optimization. Know your fluorophores, know their brightness, look at a stain index, look at your compensation and your spillover spreading. Titrate, very important. Then when you put the panel together, look at the spillover spreading, the antigen density, and then always, always be a control freak. Use your controls for gating. So I'll leave you with a slide with lots of educational resources. I think it's very important to have mentors and to have web resources. Believe it or not, I did most of my flow before there was a web, but now we have tremendous resources on the web. We have guided learning. You can use our School of Fluorescence, Lab Roots, of course. We have archived webinars that you can use some are very basic, some are very advanced. We have an Invitrogen flow cytometry panel builder that you can look at what reagents to choose for your panels, learning centers, publications. Friends don't let friends do flow cytometry alone. So I appreciate your attention. Thank you very much. And if you have any questions, please let me know.
Thank you, Ms. Oxford, for your informative presentation. We will now start the Q&A portion of the webinar. And if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, our first question is, you mentioned there are a variety of methods for optimization. What are the other methods used, and do you have a recommendation?
Yeah, that's a good question. We've been going back about those methods or back and forth about those methods for, gosh, probably 20 years now. We just did a study at Thermo Fisher and published a poster about that, and I can certainly email any participants that would like that. There are probably also talks about this on the ISAC website, International Society for the Advancement of Cytometry. My favorite method is the easy one. But of course, I want to get good data too. So I think the Peak 2 method is the one I like, uses a single bead, looking at the standard deviation and the CV. But there are a lot of methods out there. There are methods that are published by the group in Mario Roederer's lab. So if you want to take a deep dive, certainly Simon Monard did some talks on this and has published this as well, MONARD. So there's a lot of data out there, but I think my personal favorite is the Peak 2 method. The antibody capture bead method is good as well. It just takes a little bit more number crunching.
And what is the difference between compensation spillover and spillover spreading?
That's a very good question. So I really don't like the terms. Spillover sort of makes you think that there's so much fluorescence it spills over from one channel into the next. And it's really not. It's like I spoke about with FITC, you're really detecting the photons of one fluorochrome in the neighboring detector. So spillover is exactly that. You're detecting the photons of one fluorochrome in an adjacent PMT, or it doesn't even have to be adjacent. You're just detecting the photons of one fluorophore in a detector where you're going to make another measurement. Spreading is caused by photon counting error. That's just caused because of the nature of photons. If they have low energy, like teenagers, then they have a lot of spreading. It's just physics. And compensation helps us visualize that spreading, but it doesn't fix it. Until we get, new PMTs that are better detecting these photons, there's just no way around it other than good panel design.
And we're going to have time for one more question. I don't have FlowJo. Is there any other way to generate a spreading matrix?
Sure. Yeah, you can actually use the papers. There's two papers, actually. The one that I gave in the lecture is certainly available. The math in that paper is a little more complicated than I like to do by hand. There is also a paper, I'm going to try and remember it off the top of my head. It's also a Mario Roederer paper and a Marty Bigos paper. I think it's called Unraveling the Immune System 17-Color Flow Cytometry. And it has a bit of an easier math equation that looks at the spreading. And you can use that equation to generate a matrix by hand. We certainly did that before FlowJo was able to do the math. So yeah, you can do a matrix, or you can even take the matrix in either one of those papers and use it as a general guide. Certainly the value of those numbers will change instrument to instrument, but the relationship between the fluorophores is not going to change. So you can look at the ones that are an issue on any machine, and they're going to be an issue on every machine. They may be more or less of an issue, but they're still going to be ones that you're going to want to know are going to be a problem, and design your panel to begin with, knowing that is going to be an issue.
Well, I'd like to once again thank Ms. Oxford for her presentation. And before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand through April of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.